clarify the notion of resting membrane potential. Resting membrane potential is a potential difference across the plasma membrane of all living cells, whether human, animal, or plant cells. They found that there is a potential difference between the inside and outside of the plasma membrane, such that the membrane interior is negative in relation to the membrane exterior. This is found by putting the recording electrodes of voltmeter. These are the recording electrodes of voltmeter, one inside and one outside, the other outside the cell. So there will be potential difference, and we will see the voltmeter moves toward the negative charge. That's to say, there is negative charge inside the membrane in relation to the outside. The membrane interior is negative in relation to the membrane exterior. And if we go through the plasma membrane to know the causes of this potential difference, we will see that the main causes are three important structures within the plasma membrane. They are proteins. One of them is called the sodium potassium ATPase pump. This is the sodium potassium pump, and we will see its function. The other structure is the potassium channel, which is always open at the resting cell membrane. And the third structure is the fast sodium channel, which is closed at resting state of the cell. It is usually closed in resting state. So these are the three structures which are responsible for the resting membrane potential, this potential difference. How? These structures will result in uneven ion distribution inside and outside the cell. So the extracellular sodium ion concentration is much more than its intracellular concentration, while the intracellular potassium ion concentration is much more than the extracellular concentration. Why does this uneven ion distribution occurs? The sodium potassium ATPase is a protein that extrudes three sodium ions in exchange for two potassium ions. Three sodium ions exit the cell and two potassium ions enter the cell. Notice that these ions are moving against their concentration gradient. What's the meaning of moving against their concentration gradient? Because the concentration of sodium outside the cell is much more than its concentration inside the cell. So when sodium ions move outside the cell, we need to expend energy. There is energy expenditure in order to move certain substances against their concentration gradient. From lower concentration to higher concentration, we need this energy expenditure in the form of ATP. The same thing is said about the potassium ion. Potassium ions move from lower concentration to higher concentration. So these ions, when into the cell, we need to expend energy. This pump provides energy and extrude three sodium ions in exchange for two potassium ions inside the cell. But these sodium ions try to enter the cell, and they find that the sodium channel is closed. It doesn't permit them to enter. So the sodium channel is closed in resting state, while potassium channel is opened. It allows the potassium ions that enter the cell to exit from here, and they will be thrown outside the cell, while sodium ions will be accumulated outside the cell. They cannot enter the cell because the sodium channel is closed. So there will be much more positive charges accumulated outside the membrane than the positive charges inside the membrane. This process continues until we find that there are much more positive charges outside the membrane than those inside the membrane, and this potential difference is recorded in voltmeter as negative charge inside the membrane, the plasma membrane of the cell. I wish you understand the notion of resting membrane potential in this simple animation. Thank you very much.